my name is Anati Nabilah BMY TAJUDIN SHAH. You can call me Anati and my metric number is 205706. For my next part on the hands-on assessment for BMY3201, I'm going to conduct an experiment for gram staining. So let's get right onto it.
What is gram staining? It is one of the differential stain techniques which distinguishes the bacterial cells by their cell wall types. It can be either gram-positive or gram-negative. Principles: a gram-positive bacteria have a thick peptidoglycan cell wall. It does not have an outer lipid membrane lipopolysaccharide. Meanwhile, gram-negative bacteria have a thin layer of peptidoglycan cell wall. It has an outer lipid membrane lipopolysaccharide. Gram staining uses 4 different chemical reagents. The first reagent is primary stain. Its function is to impart color to all cells. Crystal violet is used to stain the cells purple. The second reagent is modern. It is used to intensify the color of primary stain. Gram's iodine is used to enhance the cell's affinity for the stain, forming a crystal violet iodine complex. The third reagent is decolorizing agent. It is to establish a color contrast in the cell. Based on the chemical composition of cell, it may or may not remove the primary stain from the entire cell or only from certain cell structures. 95% ethyl alcohol is used, which serves a dual function as a protein dehydrating agent and also a lipid solvent. The last reagent used is counterstain. It has a contrasting color than the primary stain. If the primary stain is not washed out after the decolorization, counterstain could not be absorbed by the cell and will retain the color of primary stain. Safranin is used to stain the gram-negative cells pink. Before proceeding with gram staining, a bacterial smear is prepared. For a smear from a solid medium, place 1 to 2 loops of water on the slide. Then, using a sterile loop, touch the entire loop to the culture. Emulsify the cell in the water on the slide. Thinly spread the cell suspension in the circular movement of the loop into an area approximately the size of a dime. This is to avoid the bacterial cell from clumping. Allow the slide to air dry completely. For a smear from a broth culture, the drop of water is not required. Place the loop full of bacterial suspension directly on the slide. After allowing the slide to air dry, heat fix the slide containing bacterial smear by passing it 2 to 3 times at the outer portion of Bunsen burner flame to prevent overheating. Heat fix is to ensure the cell adheres to the slide. Then allow the slide to cool. Next is gram staining process. Place the slide on a staining rack. Gently flat the smear with crystal violet and let it stand for 1 minute. Then, wash with tap water. Gently flat the smear with grams iodine mordant and let it stand for another 1 minute. After 1 minute, wash with tap water. Decolorize using 95% ethyl alcohol by adding it drop by drop until the alcohol runs almost clear, showing only a blue tinge. Wash with tap water. Finally, counterstain with safranin for 45 seconds and gently wash it with tap water. Blot dry using bibulous paper and examine the slide under oil immersion. Based on the results, the gram-positive bacteria stain violet due to the presence of a thick layer of peptidoglycan in their cell wall, which retains the color crystal violet. Meanwhile, gram-negative bacteria would stain red or pink, which is attributed to a thinner peptidoglycan cell wall. It does not retain the crystal violet during the decolorization process. Thanks for watching.